So thank you everyone for coming. Today, I'm getting the symbol to talk louder. Today, we are really excited to present to you the results of the clinical trial for our newest panel, the FilmArray Pneumonia Panel. And just a reminder that the data I'm presenting today are from assays that have not been cleared for diagnostic use, but that is in progress. So I'll just start by talking briefly about BioFire's syndromic approach. This is a philosophy that we have at BioFire. We make diagnostic tests with a symptom-driven idea. So we group probable pathogens into one large panel that's a rapid test. And the idea is that you maximize the chance of getting the right answer in a clinically relevant time frame. And we believe that syndromic testing leads to informed therapy. By testing for many pathogens at once, you increase the probability of identifying a possible agent of infection the first time, reducing guesswork and costs. This can more rapidly inform antibiotic stewardship and aid in care, as opposed to traditional testing, which is often relying on a clinician to make an educated guess about what kind of a pathogen to test for. And so we believe that this leads to the right test the first time. The technology that we've developed is called the FilmArray. This is what we call a lab in a pouch. It's a reagent cartridge that does everything needed for PCR. So cell lysis, nucleic acid purification, highly multiplexed PCR, and then a second stage PCR where we detect our analytes of interest. Using the FilmArray is very easy. The pouches come in a vacuum container. You take it out, you add it to a loading station, inject some hydration solution that hydrates all of the freeze-dried reagents, which are room temperature stable. Then you add your sample along with some sample buffer into what we call the FilmArray injection vial. That is then injected into the pouch. Everything's drawn in with vacuum, so there's no requirement for precise measuring. Then you stick your cartridge in the instrument and you are good to go. Our first test for the FilmArray platform was the respiratory panel that was released back in 2011. And over the past 7 years, we've continually developed additional assays that take this syndromic approach in mind. So we have a blood culture identification panel, one for gastrointestinal infections, meningitis, encephalitis. We have a EZ configuration of a respiratory panel that's intended for the CLIA-waived setting. We have multiple instrument platforms. In 2015, we launched a more high throughput version of our test. In 2016, an even higher throughput version called the Torch. And then most recently, in April, we submitted to the FDA our BioFire Pneumonia Panel, which we're talking about today. So the Pneumonia Panel is the largest panel that we have ever made. It's big. It's got 15 bacteria, 3 atypical bacteria, 8 viruses, and a bunch of resistance markers. I'll note that this is the first test for us. Actually, the first test, the first commercial test in the US, multiplex test that will do quantification of analytes in a multiplexed format. And so we'll talk more about that later, but just keep in mind these 15 bacteria on the left are reported with a bin value that provides some information about the quantity of organism present. There's 2 versions of this panel. The Plus version also reports Middle Eastern Respiratory Syndrome coronavirus, or MERS-CoV. In the United States, the Plus panel has a very specific intended use. It will only be used when a patient is suspected of exposure or travel history for MERS-CoV. So we talk about these 2 interchangeably sometimes because it's the exact same pouch. There's just software that hides that. So keep that in mind if we switch back and forth. If you've been following the development of the pneumonia panel, back when it was called the LRTI panel, we originally had intended to include some fungal targets, one of which was Pneumocystis cerevisiae. This organism was removed from the panel before FDA submission, and if you'd like to know more about that story, there's a poster on Saturday and also an oral presentation on Sunday. One of our collaborators from Children's Hospital of LA is going to talk about the data that we observed in the clinical trial with Pneumocystis and why that was not a great target for a broad syndromic test like this. Throughout my presentation, we have a lot of posters here at ASN this year, and I will point you to where those are as we go along. One other thing to point out, the molecular biologists got all up in everyone's business and changed Enterobacter aerogenes to Klebsiella aerogenes. So we are in the process of making those changes. So some of the slides, it's still the old way. Just one more thing to be aware of as we go along. Okay, so we have validated the pneumonia panel for use with two sample type categories. One is sputum, that includes both expectorated and induced sputum, as well as endotracheal aspirates. It's also for use with BAL specimens, including mini BALs. It is not validated for pleural fluids, bronchial brushes or washes, or nasopharyngeal swab specimens. If you're doing an NP swab, we have another test for that. Using the pneumonia panel is just as simple as all of our other BioFire tests. There's no pre-processing required. The kits come with a flocked swab, and you simply take that flocked swab and you stir it into your specimen, whether that's a liquid BAL or a really chunky and viscous sputum specimen. You use the swab to kind of mix your specimen up a little bit and put that right into the injection vial that has sample buffer, and you're ready to go. So no processing is required. It's not an issue if you have a really thick sputum specimen. It's like our other panels, about a 1 hour runtime, and it's compatible with all three of our platforms, the original FilmArray, the 2.0, and the Torch. As I mentioned, there are 15 analytes on this panel that are reported with quantification. The way that we do that is with something that we call QSM, or Quantified Standard Material. This is a synthetic DNA target that's pre-manufactured into every pouch at a known concentration. And within the pouch, the software looks at the amplification curves of this QSM at a known concentration and compares that to the amplification curves of our pathogens of interest, and then it can extrapolate an estimated copies per mil. Within the software, we take that raw calculated value and we round it into a log bin, the nearest whole log bin. So if you have a value that's 9 times 10 to the 5th, that would be rounded into the 10 to the 5th bin, and that would be what is reported. We put a cutoff on the panel, so anything that is detected, but if it's quantified below 10 to the 3.5, which is around 3,100 copies, even though it's detected, the software is going to report that as not detected. So only analytes that are present at 10 to the 3.5 or higher are reported, and then they're reported into these one log bins. So why are we binning? Why do we need to do quantification? Well, that's because determining bacterial agents responsible for pneumonia can be pretty challenging. The oral pharynx cavity is full of commensal organisms, but it's only when these commensals are present at a high level that they are suspected of causing illness. And sputum BAL samples, they often contain a lot of these organisms. So clinicians have traditionally relied upon the microbiology lab to give some sort of a quantitative or a semi-quantitative measure for many of these organisms in order to assess what is clinically relevant. So this bin reporting, it provides a context of how much and what organism is present in the sample. So if you look at our usual suspects, these guys will remain unnamed, but we've got purple, green, and blue. And if you have these organisms present in your sample, but you run a molecular test that doesn't have quantification, you might get a result that say, well, 5 organisms are present. But that doesn't really give you enough information to know which is clinically actionable. So the pneumonia panel deals with that by providing a bin value. So in this particular example, the green and the red ones were maybe there, but they were below 10 to the 3.5, so they're not even reported. These other ones were in lower bins, but this lychee, purpley thing was at 10 to the 7th or greater. And so that might be what your organism of interest is and what the clinician would want to treat for. Unfortunately, we didn't include our little cartoons on our report, but we still tried to make the report very easy to read. If you're familiar with our other tests, we usually give a 1-page report. For the pneumo panel, because it is so large, we split this out into 2 pages. The first page is just a summary. It tells you what was detected. And we have for the analytes reported with bins, we use a little bit of shading to indicate which bin they were reported in. And then the second page is the full list of all the organisms that are in the same order every time. That's to help with data entry and things like that. So, as I mentioned, we have submitted these studies to the FDA. There were a ton of studies that we did. I'm only talking to you today about the clinical trial, but I want you to be aware of all of the work we did. And we have a couple of posters here at ASM on Saturday. The analytical team from BioFire is presenting on the limit of detection and reactivity of the panel. They tested over 500 different strains to show, to fully characterize the inclusivity, exclusivity, and performance of all of these assays across broad organisms. We also have a really nice poster about precision and reproducibility. This gets at the real specifics of our bin reporting mechanism, and if you are adding quantified organisms, what sort of bin results you would expect to get. That's explained very nicely in this other poster from the analytical team. And then today in this talk, and then there's also a poster about our prospective clinical evaluation. We also did contrived and archived specimen testing for rare analytes. That's not going to be covered today. So our prospective clinical evaluation was conducted between October of 2016 and July of 2017 at 8 sites in the United States. We also used a central reference lab, MRI Global, in Florida, Melbourne, Florida. Every specimen that was enrolled in the study was taken through comprehensive comparator testing that included quantitative culture, which we'll talk about in a few slides, as well as every specimen was tested with molecular methods. That included quantitative molecular methods for our bin analytes, as well as traditional qualitative PCR and sequencing. We also did a really large medical chart abstraction. If you talk to any of the enrollers at our sites, they were pretty annoyed with us because we were collecting about 2 weeks of data on every subject that was enrolled because we really wanted to be able to do some retrospective analyses, which I'll show at the end, to understand what was going on with these patients and where the pneumonia panel could have had an impact in their care. But mostly today we'll be talking about the comparator testing that was done to look at performance. The enrollment criteria for the study were pretty simple. The specimen was just a residual lower respiratory tract specimen, so sputums, ETAs, BALs, or mini BALs, that was submitted to the lab for culture. So it had to have been sent to the lab for culture workup, and there was culture results available. Specimens were not centrifuged or pre-treated in any way. They were collected from subjects who had not previously been enrolled in the study, so all unique patients. And we tried to collect the specimens as fresh as possible, so within 24 hours of collection from the patient, stored at 4 degrees, never frozen, so they could be enrolled and tested very quickly, and then shipped overnight to our central reference lab. We excluded any subjects that had cystic fibrosis or active TB. People who had latent TB, they may have been enrolled if it was not part of their active medical chart. So our subject demographics, we ended up in the end enrolling over 1,600 specimens, close to 1,700, about equal numbers of BAL and sputum, from slightly more male than female patients. Typically, I think more male patients happen to be hospitalized, which might lead to that bias. I want to point out that we also had 2 pediatric institutions in our trial, and so we had a really nice representation of pediatric patients in our study. Not so many BALs being done on peds, but we did have about 30% of sputum specimens coming from subjects under the age of 18. We also were enrolling from multiple care settings, so not just hospitalized patients, but about 20% of patients, when they initially presented and the specimen was collected, they were either in the ED or coming in from some kind of an outpatient setting. And this might have been long-term care facilities or other non-hospitalized patient population. So just to talk briefly about our system performance, this is a measure of just how often did the test complete successfully, and that was over 98%. The denominator here is 1798. We only had 1,600 specimens, but we were doing daily controls and things like that, so that's how we get to almost 1,800 actual FilmArray tests. And of those, there were only 2 where there were software errors or instrument errors, so 99.9% instrument success rate, which is great. And then there were 32 tests where there was an invalid pouch control, so that's a very low control failure rate, under 2%. We're very happy with those results. When we look at just what was observed by the pneumonia panel, and this is all detections, bacteria, viruses, and atypical bacteria, we can see in BAL specimens that of the 846 BAL specimens, there were 413, or almost 50%, that had at least one analyte detected. And there were even, in this case, in this sample set, there was one that had 6 or more analytes. So that's kind of expected in these pneumonia specimens. They're full of a lot of organisms. In sputum, there's even more. So 72% of sputum specimens were positive for at least one analyte, with many more having a higher number of co-detections. So highly positive sample matrix. So in order to determine our performance metrics, we were comparing the FilmArray to different reference methods. For atypical bacteria and viruses, this was 2 PCRs followed by sequencing. This is pretty standard. It's what we do for most of our other panels, and other companies do as well, to demonstrate performance. For antimicrobial resistance genes, we did PCR and sequencing direct from the specimen, but then we also did testing on any isolates that were recovered by culture. We did AST for phenotypic resistance, and then we also did PCR and sequencing for the gene of interest from these isolates. This was done for informational purposes. And finally, for our bacteria that are reported with bin values, our primary method for sensitivity and specificity was a comprehensive quantitative culture, and our second method for looking at bin performance was a quantitative molecular method, followed by PCR and sequencing. So we'll get right into the data here. This is the giant performance table for viruses compared to what we're calling conventional PCR and sequencing. You can see all of the virus analytes listed on the left. One thing I'll mention, for MERS coronavirus, we assumed all specimens were negative because MERS was not circulating in the US at the time of the test, so there was no PCR comparator there. But we see there were no detections, which is great, very specific. We validated MERS with some different testing we're not showing today. So what I want to point out is that the sensitivity and specificity is really excellent for most analytes. There are a couple of areas where we're not above 95%. So for example, for influenza B in BAL, there was 1 missed detection out of 6. So that gives you a performance hit when you've got a false negative there. And then for adenovirus and coronavirus, we have a couple false negatives and false positives. When we went back and looked very closely at the data, what we saw is that in particular for adenovirus and sputum and coronavirus, there's kind of this long tail of low-level detection that's in these specimens. And the FilmArray assay and the comparator assay had equivalent limits of detection. And so when you're trying to measure an analyte presence that's below the limit of detection, you sometimes get a positive or a negative. And so that kind of leads to those discrepant false positive and false negative results. What's kind of interesting is that in BAL, where we have 100% concordance with adenovirus, both sensitivity and specificity, when we look at the cycle values for adenovirus in BAL, it's much higher. So there's something biologically going on, maybe, that in BAL specimens, adenovirus is just at a higher level, whereas in sputum, it was typically at a low level, and that was leading to those discrepant results. Here's the performance for our 3 atypical bacteria. Unfortunately, there just were not that many of them encountered in the trial, so we don't have a lot of numbers. So the performance for these organisms was mainly demonstrated through archive specimens and contrived testing. So getting into these 15 bacteria that are reported with bins, how did we do the comparator testing there? Well, we did a very comprehensive quantitative culture. This was all done at MRI Global in Florida. Our sites were FedExing specimens overnight, refrigerated temperature, 3 days a week. We paid a lot of money to FedEx. We got a lot of experience with the shipping facility in Tennessee that likes to lose packages. But overall, it worked pretty well. So specimens arrived at the reference lab, and they were always plated for workup within 2 days of collection from the subject. So as fresh as possible and always at refrigerated temperature. The first thing they did upon arrival was do a gram stain, and then they plated these samples for quantitative culture. Each specimen was plated at 10 microliters and 1 microliter, as well as a 10 and 1 microliter plating of a 1:100 dilution across 4 different plates: blood, chocolate, McConkey, and CNA augers. So that's 16 plates for every specimen. Specimens were read at both 24 and 48 hours, and technicians looked for any unique-looking colony morphology. They made a note of it, they counted it, they set it up for ID on the Vitek, and they, when appropriate, they then did AST testing and made glycerol stocks for every unique-looking morphology. In the end, this was over 25,000 plates. That's a lot of plates. This isn't an actual picture from them. This is from the internet, but I think it represents what they went through. They had these big walk-in 35-degree incubators that they had racks of plates on. It was pretty impressive. So that's how we did our comprehensive quantitative culture. Before the quantitative culture study started, we did a reproducibility experiment where we sent 32 clinical specimens to the reference lab. Each specimen was separated into 9 different aliquots and plated by 6 randomized technicians. And the question is, if you have one specimen that is independently worked up 9 different times, what does the variation look like? And I'll let you go find poster number 341 on Saturday to find the answer to that. A spoiler alert, it's highly variable. For the first measure of bacterial performance, we're doing a qualitative comparison. So for this, we're just looking at positive, negative. And for this interpretation, culture comparator is considered positive if it was identified by Vitek 2 from the isolate cultured from the specimen, and if the colony count was enumerated at greater than 10 to the 3.5. So remember, that matches the FilmArray cutoff that we're using, which is also 10 to the 3.5. And then the pneumonia panel is considered positive if it was detected and reported at 10 to the 4th, in that 10 to the 4th bin or higher. So that gives us our detected, not detected metrics for this next slide. So here is our bacterial assay performance in BAL alone. And what you can see is that we had very high sensitivity and specificity numbers that are greater than 90%. So this was really nice. We do have a couple of false negatives. In all 840 some BAL specimens that were tested, there were only 3 false negative results. One of those, the BioFire panel did detect the analyte, but it was below 10 to the 3.5, so it was reported as not detected. So that is a true miss. It's a false negative. One was also completely missed by BioFire, but the molecular comparator picked it up. So again, that's really a false negative. And then the last one, both BioFire and our molecular comparator missed it, but standard of care was concordant with the culture comparator method. So we think that's also a true miss. So three total false negatives. So our specificity numbers look really nice, but 90% looks good. But if you delve into it, that's actually 90% of a very large number. There are a lot of false positive results, particularly for organisms that are fastidious, like Haemophilus and Moraxella. Interestingly, you'll notice for Moraxella, there were no BAL specimens where Moraxella was recovered by culture in the central reference lab, because this is a very finicky organism. But we did a lot of discrepancy investigation on these false positives, and of the 326 false positives, a third of them were actually enumerated. They were cultured and enumerated, but they were below the 10 to the 3.5 cutoff, so they were considered negative. Of the remaining 2/3, they were completely missed by culture, but they are detected by our other molecular method, so they are true positives in a sense. 3 were missed by both comparators, but were concordant with standard of care, and then there were 2 unresolved. So in this huge data set, basically there's just 2 false positives that we can't explain, which we think is pretty good. Sputum is very much the same story, but with many more detections. Again, sensitivity is very good. Here we have 13 false negatives. 5, again, were the BioFire detecting it below 10 to the 3.5. 2 missed completely by BioFire, but evidenced by molecular method. And then there were 5 instances where the central reference lab gave an ID, and when we did additional molecular testing of those isolates that were shipped to us, they did not match the original ID from the culture. So these could have been just issues with Vitek 2 giving a misidentification. I think one of them turned out to be a Hafnia. There were 2 Pseudomonas fluorescens and a couple of other random things. And so it's not clear why the reference lab identified them originally the way they did. But by molecular methods, they were not what the reference lab said they were. And then there was one instance of a specimen swap. It's very clearly a specimen swap, but all the paperwork says that it's not, so we have to take it as a false negative. It's fine. Again, on the false positive side, our problem organisms, Haemophilus and Moraxella, are a major driver of this. There's 547 false positives in this data set. A fifth of them, again, were enumerated by culture, but present below that cutoff. The remaining 80% were detected by the molecular method. There were 9 missed by both comparators, but were reported by standard of care. And then again, only 2 unresolved. So here's another poster you might want to look at. On Saturday, poster 230 is our R&D team put together a really comprehensive poster explaining how we did all of our discrepancy analysis. That's both for these bacterial targets and the viruses that I presented previously. So why is culture missing so often? It was a surprise to us that the culture reference method was so discordant with our FilmArray results. And that's because when we did all of our validation testing in-house and we grew organisms in our BioFire laboratories and we counted them with colony counts, CFU per mil, this is an aggregate plot of all organisms enumerated by CFU per mil versus FilmArray. And what you can see is that it's shifted maybe about a log difference, but it's still a very linear curve. But when you think about our lab culture at BioFire, it's sort of the most ideal conditions you can think of, like a greenhouse, right? We have these organisms that are grown in pure culture, they're harvested at log phase, they're in a nutrient-rich media, and basically it's the most ideal conditions for every genome to create a colony-forming unit. And so it doesn't get better than that. But in our clinical data, we've got biology going on. So here are the same type of a scatter plot of the FilmArray quantified raw value versus the reference culture value. And you can see that it's just a big smear. It's not linear. And that's because clinical specimens, like it's the jungle. So you've got all these biological factors, patients on antibiotics, patients who've been sick for various amounts of time, different immune response, many different factors that could result in loss of viability of these organisms and why they're not being cultured out. So that was a bigger discrepancy than we were expecting. So in order to validate the bin result of the FilmArray, the quantification value, we needed a better comparator than quantitative culture. And for that, I back up just a second. We have another poster where I told you we had a huge medical chart abstraction for each of these patients. So our biomath team at BioFire did some machine learning algorithms to try and see if they could find clinical factors in the patient chart that would predict whether the organisms would be recovered from their culture at levels that matched molecular or not. And so if you want to check out poster 266, you'll see the machine learning. I'm not educated enough to talk about machine learning, but it's all on that poster, and they're here to explain it. Okay, so we needed a quantitative molecular method in order to validate our bin approach. When we looked at trying to do 15 PCR reactions with standard curves across 1,600 specimens, we realized we would need about 100 thermocyclers and a team of RAs in a year to do it, and we had to come up with a better method. So our R&D team developed a new technology that used PCR followed by next-generation sequencing to do really multiplexed and highly multiplexed quantitative analysis. And so basically the overall picture of how that happens is that we take our specimens, they're spiked with a known quantity of that QSM, the same quantified standard material that's in the FilmArray pouch. The specimens are extracted like normal, and then we do a highly multiplexed PCR. This is with targets that are independent of the FilmArray targets. So they're independent PCRs. They are then tagged with DNA barcodes for next-gen sequencing, pooled together, and then put through a next-gen sequencing pipeline. Our biomath team and software team then developed custom software to analyze all this next-generation sequencing, and the end result is that you get a quantitative analysis of these next-gen sequencing reads. And when we do that and compare the quantitative molecular method to the FilmArray unbin value, what you see is really nice correlation. So FilmArray quantification is matching very nicely with a completely independent molecular quantification method. So these scatter plots look really nice, but we had to try and come up with a way of expressing these in sensitivity and specificity. I'm not going to go into too much detail about how we did this, but basically we tried to come up with some bin performance tables that show the percentage of time that a FilmArray bin is concordant with a range of quantitative molecular testing, and we can talk more about this individually later. But the bottom line is that by these methods, about 86% of the time, the FilmArray bin is concordant with the quantitative molecular method. And this is an analysis that we came up with based on some FDA guidances. FDA is reviewing this. We don't know if they're going to agree with this sort of approach, but we think it's a nice way of putting those scatter plots into a table format that's a little bit understandable. Another way that we can think of looking at the bin data is with these stacked area plots. So in these plots, what we're looking at, if you can see, we have on the bottom, we have our quantitative comparator value, and then these shaded areas indicate the proportion of FilmArray results that are expected to be within a particular bin. So if your quantitative comparator method gives you a value of 10 to the 5th, which is right in the middle of the 10 to the 5th bin for FilmArray, which spans 10 to the 4.5 to 10 to the 5.5, you would expect that almost all of your FilmArray results would be in the 10 to the 5th bin. If your quantitative comparator value gives you a value that's on the edge of the bin, say 10 to the 4.5, if you follow that up, you would expect that half your results are in the 10 to the 5th bin and half are in the 10 to the 4th bin. So this is kind of a graphical way of trying to visually represent the bin concordance of FilmArray and the comparator method. So again, keep in mind, this is what the model looks like. And then if we look at our actual clinical data for BAL and sputum specimens, the stacked area plots are pretty similar to the model. So this is a way to kind of visualize that FilmArray bin reporting is pretty close to our quantitative comparator method. And then sputum is not. I'm sorry, culture is not in sputum or BAL. And so that, again, is just because of the differences with the culture-based method. So finally, I want to talk about our performance of antimicrobial resistance genes. Again, this is looking on a per specimen basis when an applicable organism is reported by FilmArray and an applicable antibiotic resistance gene is reported, how often that is concordant with PCR from that same specimen. And basically, except for MEK and MREJ, we don't have a whole lot of observations, but the sensitivity and specificity looks pretty good. We do have a couple of false negatives and false positives, and that is due to some results that are near or below LOD. We did a lot of discrepancy investigation here. We think that a lot of this is coming from low-level background flora, so carbopentamases may be carried in organisms that are not detected by the FilmArray panel, but are present in the respiratory tract at a low level. And then with Staph aureus and MRSA, this is a common problem with mixed infections where you have sensitive and resistant organisms and empty cassette organisms and MEK floating around in Staph epi. And so in that soup, you just kind of get some different combinations that lead to some discordant results. So what I hope I've showed you so far is that the FilmArray Pneumonia Panel, when testing clinical specimens, it's doing what we think it's doing. It's accurately counting genomes. It's accurately detecting nucleic acid. But one question is, what does that mean on an individual patient specimen level? So to do that, we've turned to some hypothetical outcomes analysis. This has been performed by our collaborators at the Medical College of Wisconsin. And what they've done is they've taken that huge case report form that we collected, and they're doing a really detailed analysis on a subject by subject basis. So for this analysis that they've just started doing, they're initially only looking at BAL and only adult patients and only inpatients. So that's the subset of data. They're looking at a composite standard of care result. So anything that was ordered for the patient, whether that was bacterial culture, that could have been reported quant, semi-quant, or qualitatively, whatever the site happened to be doing, that's what's in the medical record. And then any virus or atypical bacterial nucleic acid tests that were ordered just based on what the physician had ordered at the time. And then this in-depth chart review, they're looking at specific symptoms and comorbidities, antibiotic start and stop dates. We collected that for a 2-week time period. the time to result for the standard of care results, and then their discharge diagnosis either at day 14, the discharge diagnosis or what the diagnosis was at day 14 when we stopped collecting data, whether it was infectious or non-infectious. And the idea was to try and understand how does the Pneumo panel result compare to standard of care, and where could it have made a potential impact? So there are 3 cases that I would like to highlight. Case number 1, this was a 32-year-old male with progressive right facial droop and right upper and lower extremity weakness. They were eventually found to have a glioblastoma and had surgery. Patient was intubated and developed a low-grade fever with no leukocytosis. Chest x-rays was not super specific and informative, and so the question is, what would the physician do? In this case, the physicians decided not to start antibiotics. The patient continued to have a low-grade temperature, but still no leukocytosis and nothing indicative in the chest x-ray that was of concern. 48 hours later, the standard of care culture grew Haemophilus and strep pneumo mixed with normal oral flora, and they were started on ceftriaxone. So this subject was enrolled in the study. We had a FilmArray Pneumo panel result for them, and it reported H. flu at 10 to the 7th, strep pneumo at 10 to the 7th, and E. coli at 10 to the 5th. So if this panel had, if this result had been available to the clinician, it is possible that this patient could have been put on appropriate antibiotics 2 days sooner based on that result. In the second case, this is a 68-year-old male with lung cancer presenting to the hospital in the middle of winter with fever and some other respiratory symptoms. It's slight leukocytosis and chest x-ray without any clear infiltrate. So would this be infectious? Should you start him on antibiotics? Because this patient had lung cancer, the physicians, I guess, were being cautious and started them on Piptazo. But 48 hours later, the respiratory culture was negative, and so the antibiotic was discontinued. And there was no other respiratory workup completed, and the patient eventually improved, but the patient was admitted. The Pneumo panel reported influenza A and was negative for all bacterial targets. And looking through the medical record, influenza A was not on the differential at all. No one ordered any testing for influenza A. So if they'd had the Pneumo panel result, this could have enabled appropriate antiviral therapy, and also that could have reduced the duration and severity of symptoms. But also, this patient was admitted and had influenza, and so this is a really important consideration for infection control to prevent transmission, and an opportunity that was missed. So we think this really highlights the importance of our syndromic approach of including viruses on this type of a panel where they are not otherwise suspected. And finally, case number 3, this is a 62-year-old female with a liver kidney transplant. They developed respiratory failure. Again, no leukocytosis. There was something going on the chest x-ray, but it was attributed to diuretics, continued to decline, and was intubated 2 days later. The bronchoscopy revealed, the gram stain revealed gram-positive rods and cocci, and they were started on vancomycin. But 48 hours later, the culture reported normal oral flora, and vancomycin was discontinued. The FilmArray panel reported methicillin-susceptible Staph aureus. So reported Staph aureus and no MREJ or MAC-A at 10 to the 5th. So you could ask if it was appropriate to discontinue all antibiotics. There wasn't enough data in the medical record to know if this patient relapsed or if there were further consequence of this. But the important point, I think, is that the Pneumo panel could have enabled a targeted de-escalation or use of a more specific therapy right from the start. So these are 3 examples from our actual data where we think the Pneumo panel could have made a pretty significant impact in patient care. So in conclusion, what I hope I've shown you is that our pneumonia panel is a very specific test when compared to culture methods. However, there are some specificity considerations relative to culture, and that's just due to the fundamental differences between molecular methods and culture methods. It's a very sensitive test. Our false negatives were there were very few false negatives. In our qualitative analytes, it was attributed to things just being below LOD or a couple of misidentifications by the reference method. And I think I've shown that the binned bacterial result assigned by the panel is highly concordant with that independent quantitative next-gen sequencing, which is a totally independent molecular method. But it's important to remember the test consistently reports bacteria at higher levels than reference culture will, just again due to those fundamental differences. And we believe that the syndromic approach to the pneumonia panel has the potential to improve patient outcomes through that retrospective chart review. So there's a lot of people to thank. We had 8 sites participating in the collection of specimens and the generation of all of that case report form data, a team of people working at MRI Global for the Central Reference Lab. And this might be a little unusual in an industry talk, but I really want to point out the huge team at BioFire that worked on this panel. It's one of the most complicated things we've done. Certainly the most complicated project in a long time. This was a collaboration as well with our colleagues at bioMérieux in Grenoble and Verneuil in France. And we are really proud of this panel, and we hope that we can get FDA clearance soon and bring it to you. And I'll leave you with the list of posters and the presentation that is being presented at this meeting if you'd like further info, and I'll open it up for questions. Yes.
I'm sure I missed this earlier on, but was there a role for the gram stain to evaluate the quality of the specimen, and was that part of the evaluation?
So it was not part of our performance evaluation, but so we were collecting gram stain that was in the medical record standard of care. But as I mentioned, the first thing that the reference lab did when they received a specimen was a gram stain. So we do have gram stain data, and we do have quality scores for every specimen so that we can go back and look at if there's an influence of quality score on performance. And I don't have that data included, but I can just tell you in general, about half the sputum specimens would have been considered low quality. But the ability of FilmArray to detect nucleic acid compared to the quantitative molecular method and compared to culture was not affected by a low quality score. So we do have that data, but we have not found that it was a significant driver of performance. Okay, well, everyone enjoy ASM, and thank you for coming.